Yes, we have seen that after making amino acids, after feeding these cells with enough amino acids, then the next stage is transcription. Now with transcription, this is the process by which a base sequence on DNA or of DNA that represent a gene of interest is converted into a base sequence of mRNA strand. It is as simple as that. Like I told you, this is your DNA, and the DNA is double helix. Now, of the two double helix, one is a template strand, and the other one is a complementary strand. And not all strands are copied, but it is this template strand that will be copied during protein synthesis, but in DNA replication, all strands obviously are copied. So, you have DNA, and DNA has genes, maybe from here to here. This is what codes for maybe hormone A, okay? Hormone A. And maybe from here to here, it codes for maybe hemoglobin. Maybe from here to here, it codes for maybe enzyme A. Now, the body, in this particular case, it wants to synthesize enzyme A. It is this segment here, okay? It is this segment here that will be copied. It will have, yes, you have flags that will switch on that gene, that particular gene. You have some clues that will tell that it is this gene that should be synthesized. So you have clues that will open up only that gene, and it's that gene that will be transcribed. Now the process of converting the base sequence, maybe you have A here, you have C here, you have G here, you have T, and here you have A, and here you have A, and finally you have G. So this is the base sequence that represents this gene on DNA. Now the process by which this base sequence on DNA is converted into a base sequence of mRNA. And if it's mRNA, this one is going to be U, this one is going to be G, this one is going to be C, this one is going to be A, this one is going to be U, and finally C. So that process of converting this base sequence on DNA into a sequence of bases on mRNA strand is what we call transcription. So if you know what it is, then everything now becomes easy. If you remember DNA replication, then still everything becomes easy. So now what happens during transcription? Now I've talked about this. The section of DNA and this region is called ancestron. You know, you have DNA and that segment, okay, of DNA that represents a gene, okay, is known as ancestron. So in other words, ancestron is an alternative form of a gene. No, alternative form of the word gene, not alternative form of a gene. That's an area. So if you want to be more fancy, you can say cistron instead of gene. That one is cool. Now what happens in DNA replication? Okay, in DNA replication, we saw that the double helix unwinds to expose the nucleotides. And then DNA primase, it comes and adds primers Okay? It adds primers to show where to start from. And then DNA polymerase adds these nucleotides. It is almost similar process. But now here, yes, it begins with unwinding of the DNA. Okay? Like how you can see this. The DNA unwinds. And which enzyme is responsible? It is still DNA helicase. Okay? And then after unwinding of the DNA, or after the DNA has been unwound, then RNA primase, it will come and insert nucleotides on where to begin from. Okay? And like I told you, not all strands of DNA are copied. It is the template strand that will be copied. So if this is the template strand and this was the complementary strand, we shall copy only this. And this template strand is going to be used as a template for synthesis of the new mRNA molecules. Now after adding your primer, now instead of DNA polymerase, now the DNA polymerase, for it is responsible for formation of DNA. Okay? But now, for us, we are interested in forming RNA. Now, it is RNA polymerase. Okay? RNA polymerase that will now come and bind, in fact, the whole of this. It is RNA polymerase. So it is the one which will come and bind to that specific segment that represents a gene and then starts adding the free nucleotides in a complementary manner to form one single strand of mRNA. To form one single strand of mRNA. And addition is complementary. Let's see how complementary it is. So RNA, it moves along the DNA cistron, adding complementary nucleotides together. Yes? Okay, you're welcome. So, we said the RNA after unwinding, and then the RNA, you've opened that template strand, you've established where the gene begins from, then the RNA polymerase to come and bind and starts to add these nucleotides together. Okay? Yes. And the addition is complementary. It's complementary. Okay? Yes, don't forget the other complementary rule. Where there is C, you add G. Where there is G, you add there C. Where there is T on DNA, this is DNA, and here you are forming your new mRNA strand. So where there is T, you add there A. And where there is A, now you cannot add T. Because RNA, we saw one of the differences is that RNA has no thiamine. But thiamine is replaced with base uracil. So we shall add there uracil. Okay? And in such a complementary manner. So you can have a look at this. You see, here there was C on DNA, and we are adding G. There was A, we are adding U, C, G, A, U, T, A, so and so on. Okay? And you'll be able to synthesize a template strand of DNA. Now within a gene, you have promoter regions. Now those promoter regions, it's where actually the gene begins from. Okay? And you have regions where the no-sense regions. You have regions where the gene end from. So it is going to start from here, from this promoter region, and adds nucleotides. Okay? Until when you reach the end of the gene. And when you reach the end of the gene, you will terminate the process. The process of adding nucleotide is going to be terminated, and you form one single strand of mRNA. Now, addition of nucleotides is an active process which involves energy, and because you know that DNA is highly negatively charged, then you also need inorganic ions such as magnesium ions to stabilize the structure of DNA. Okay? So you may find an objective question which wants the role of magnesium ions, or which the question is like, which of the following plays an important role in DNA replication or in protein synthesis, stuff like that. And they say A, magnesium ions, B, sodium ions, C, calcium ions, D, chloride ions. So the answer here is magnesium ions. Why? Because you know that this DNA is highly negatively charged. It's negatively charged in a way that it's made up of a phosphate sugar backbone. So if this is a phosphate, then this is a sugar, and then this is another phosphate, then this is another sugar. Now this phosphate, they are formed from phosphoric acid. Okay? Phosphoric acid, it is this H3PO4. So this phosphoric acid, after condensing, you remove one H, you remain with hydrogen phosphate. Okay? So this can lose these hydrogen ions, and you remain with highly negatively charged phosphate groups. So now these magnesium ions, they play an important part as far as stabilizing these negative charges on DNA is concerned. So that these enzymes, the RNA polymerase and the DNA polymerase, can do their processes without interruptions. Now at the end of transcription, the RNA polymerase recognizes the stop codon and becomes detached from the gene region. And then you form a single strand of mRNA, this very one. You form a single strand of mRNA. Now, this single strand of mRNA, like I told you, even within a gene, there are some parts of a gene that do not code for any amino acid. And then there are other parts within a gene that code for certain amino acid. For example, you may find from here to here, it doesn't code for any amino acid. And then from here to here, that's what can code for amino acid. Let me represent it with yellow. Okay? And then from there to here, it doesn't code for any amino acid. And then from here to around here, it codes. And then this doesn't code for any amino acid or any proteins. But from now here to here, it codes for proteins. Oh my god, I wasn't supposed to wrap. But let me sketch again. Okay, let me sketch again. So you have this, you have this, and then you have this. So this part here, it doesn't code for any protein. This part here, it doesn't code. It is non-coding region, and this one is also a non-coding region. But now this is the coding region, even this one, even this one, even this one. Now these non-coding regions within DNA are known as introns. Yeah, it's okay. Yes, so we have said that these non-coding regions, they are known as introns. Okay? And then these coding regions, these ones, here, here, and here, these coding regions, they are known as exons. Not axon, but exon. Or exon, whatever. This one. Axon is this, axon. But this is exon or axon. So this is the spelling. The spellings are different. So the coding regions are known as exon, and the non-coding regions are known as introns. Now there is no need of leaving this mRNA to move or to diffuse to the cytoplasm for translation when it still has, I think, these introns, these non-coding regions. They are useless. These non-coding regions, they are useless. So what happens? You have your enzyme that will come and split this mRNA molecule. It split it from here and here to remove this non-coding region, and then you form one continuous strand that can code. And then you do the same here, you split from here, and then you bind this part to this part. You form another continuous region. Then you come and remove this, and you also remove this, and then you join this to this, and you form another continuous strand of mRNA. Now this is the processed mRNA that can now code for a protein. Now this process of removing these non-coding regions called intron, it is known as splicing. Okay? And this will be the complete mRNA molecule, okay, that can now diffuse to the cytoplasm for translation. So now after forming this, you have their transport proteins within the nucleus, many of them. So you bind this mRNA molecule to a transport protein within the nucleus, and then the transport protein, it will diffuse out of the nucleus through this nuclear pore, okay, and take this strand of mRNA to the ribosome for translation to take place. So in case there are queries, feel free guys to ask. So somebody is asking that, is splicing, or does splicing occur on the DNA, or it occurs on the mRNA that has already been transcribed? And the answer is it occurs on the mRNA that has already been formed. So this is what I was illustrating. So you have, this is the new mRNA strand that has been transcribed, and the strand has the coding regions called exons and the non-coding regions called introns. So splicing involves removal of these introns. You come and cut from here and here, you remove this segment, and then you join the two. I think that is ligase enzyme, it does that. Then you also come, cut from here and here, then you join these two, and you form one complete strand of mRNA. So basically that's what splicing is.